Ladies and gentlemen, today is my distinct pleasure to introduce our speaker today in this webinar, Dr. Udeme Ekong from the Georgetown University, USA. So Dr. Ekong is one of the leading experts in pediatric hepatology and liver transplantations with research focus on immune-mediated liver diseases, including autoimmune and autoimmune hepatitis. So Dr. Ekong serves as a PI on many research projects, also on NIH-sponsored clinical studies, examining immune mechanisms and long-term outcomes in pediatric liver transplant recipients. Her work has been published in many respected journals, Journal Immunology, Frontiers Immunology, Pediatric Transplantations, and she has also contributed to international clinical guidelines in pediatric hepatology. So this afternoon here Italian time, Dr. Ekong will give us her presentations entitled Human Endogenous Retroviruses in Inflammatory Liver Disease, whether something to consider or nothing doing. So thank you again, Dr. Ekong. So time is yours.
All right. Thank you so very much for the very kind introduction. Let me share my slides and then we can get started. Just 1 second. Please. And can you see my slides? Can you all see my slides? Sorry, I can't hear anyone.
Yes, we can see your slide.
Yeah.
If you make presentation mode, it would be even better.
If I do it at presentation mode, you will see my presentation mode.
Can you do it like we did before?
Isn't that what it is right now?
No, we still see to maybe go to play from start. Just close the presenter view.
Okay.
Okay.
Hold on 1 second. Okay. So, is that better? That should really just show the regular slides now. It's not.
No, we still see the your side of the presentation, I think.
Wait.
Okay.
I'm just doing the same thing we did just before. I'm doing the same thing we've done.
Okay.
That's interesting. Let's do it. Let's do this. Let's try that one more time. Is that better?
We haven't seen your slide yet.
Okay. Okay, I think I know what I need to do. Hold on. I think what I just need to do is, first of all, just get it. And then I lose.
Okay.
Is that better?
Perfect. Yes, this is
that's wonderful.
Thank
Okay.
you.
Sorry about all that. So, good morning, everyone, and thank you so much for the kind invitation to speak today. I'm going to be talking on human endogenous retroviruses and inflammatory liver diseases. Let's see if I can minimize that some. Okay. So this is the outline of the talk I'm going to be doing today. I'm going to start off with a background of post-transplant autoimmune hepatitis. And as had been alluded to earlier on, the main areas of my interest in our research has really been in autoimmune hepatitis that we see after liver transplant, as well as autoimmune hepatitis. And so, because I don't really know the background of everyone on this talk, I'm just going to start off with a brief background of what this condition is and why it is important to pediatric transplant hepatologists like myself in terms of looking at it from a context of long-term outcomes. And this is because we know that in North America, a child who gets a liver transplant in North America is getting a liver transplant, is transplanted in infancy. And so our commitment to these families is that you're going to live with that same liver till you become a grandmother or grandfather at 80 years of age. And so things that could negatively impact long-term outcomes are very important to people like me who look after children. And then the second part of my talk is going to veer into the work that we're doing in a translational science laboratory, specifically on human endogenous retroviruses. I'm going to touch on some of the collaboration we have, which is still in infancy, with investigators and scientists at the National Institutes of Health, or the NIH here, as well as some other ongoing work that we have with an investigator or scientist at the Cancer Center here at Georgetown. So as a background, post-liver transplant autoimmune hepatitis is a poorly understood cause of late graft dysfunction. It shares biochemical and histological features with autoimmune hepatitis that we see in the native liver. And if you look at reports from pediatric liver transplant centers, they quote a frequency of about 5 to 10%. And then also when you look at reports from adult liver transplant centers, the frequency quoted is about 3 to 5%. But importantly, its cause and underlying pathogenesis remains unclear. And so these are usually individuals who are transplanted for diseases that we generally do not think have an underlying immune-mediated basis. And I've listed some examples on the slide. And most of these cases present over a year after transplant. And the things we use to make this diagnosis is very similar to the way we make a diagnosis of autoimmune hepatitis in the native liver. And so that's the presence of autoantibodies, elevated serum immunoglobulin G, and a portal-based inflammation on a liver biopsy that's predominantly lymphocytes and plasma cells that have that characteristic finding that pathologists call interface hepatitis. And lastly, there's usually the absence of any other thing to explain the cause of the allograft dysfunction. And so this is just a H&E slide that tries to depict what autoimmune hepatitis looks like in the transplanted allograft. So again, you have here a portal-based predominant inflammation that's usually made up of plasma cells and lymphocytes, and that characteristic extension beyond the limiting plate of the portal tract in a manner known as interface hepatitis. And so we asked ourselves a question, what are the long-term consequences of a child developing autoimmune hepatitis in their transplanted liver allograft? And to try and answer this question, we used data from 5 pediatric liver transplant centers in the UK and in North America. 4 or 5 of these centers were large centers, and one of these centers was just a smaller center doing about 10 livers a year, and that was Yale. So these were Birmingham Children's in the UK, Hospital for Sick Kids in Toronto, Canada, Lurie Children's, Northwestern in Chicago, Columbia Presbyterian in New York, and then, as I said, Yale New Haven in Connecticut. What we did was we looked at the databases in these 5 centers and identified all those children who had a diagnosis of autoimmune hepatitis, and then looked at their outcomes from the time of diagnosis to either death, retransplantation, or transfer of care to an adult institution. We were working with a denominator of about 1,800 odd children who had undergone an isolated liver transplant, and 31 of them had a diagnosis of autoimmune hepatitis. And when we look at the median duration from transplant at the time they developed autoimmune hepatitis, was about 5 years. We had complete follow-up data in 29 of these children. And in fact, the follow-up from the time of diagnosis was as far as 15 years from diagnosis. And so just to share a couple of pertinent results that we observed. At the time of last follow-up, what was very striking to us was that many of these patients were still receiving quite significant immunosuppression. So about 2/3 of them were still on steroids, 3/4 were still on an antimetabolite, and 7% were on an mTOR inhibitor. And in spite of this, they continued to have liver perturbation as evidenced by abnormal liver tests that was observed in 38% of these patients. At the time of manuscript publication, 14% of these patients developed progressive liver disease, and 10% were relisted and were retransplanted for an indication of chronic rejection and portal hypertension. And so just to summarize the findings we thought were pertinent in this study, we saw the observation of continued perturbation of the liver. And these raised the question in our mind, are there other pathways that are driving disease pathogenesis that we're not addressing in the treatment paradigms that we have presently? Also, as a consequence of this persistent liver perturbation, there was development of progressive liver disease necessitating retransplantation. And this again underscored the inadequacy of the present treatment options that we have. And lastly, graft loss resulted in a need for retransplantation, raising the question in our mind, is it possible to identify those patients who could be at risk for developing autoimmune hepatitis even before it appears? And so our lab, our translational research lab, has really focused a lot on regulatory T cells, rightly or wrongly. And that's really because we know Tregs play a role in maintaining tolerance to self-antigens. They're known to curb autoimmunity. And we know that when they are defective, this results in a breakdown of self-tolerance. And my lab also, in the last few years, has really veered into on trying to study human endogenous retroviruses, or HERVs. And these HERVs usually form about 8% of the human genome. And they usually lack function and expression as a result of restrictive epigenetic mutations or genetic mutations, even though some of them have been shown to exert some key physiological functions. But if you do look in the autoimmune literature, specifically the diabetes literature, multiple sclerosis literature, rheumatologic literature, there are several reports of HERVs being expressed in pathological processes in these autoimmune diseases. And this has led investigators to hypothesize that HERVs may trigger autoimmune diseases through various effects, including immune dysregulation. But the exact role of doing this of them triggering autoimmunity remains unclear. Now, our lab has reported or demonstrated that the envelope protein of HERV1 actually induces endoplasmic reticulum stress with resulting unfolded protein response activation in Tregs of children with autoimmune hepatitis, as well as children with autoimmune hepatitis. Now, I'm not going to go into detail on this paper because this is obviously published work, but suffice to say, just to one slide or two slides on the endoplasmic reticulum, the UPR system. So this is a three-pronged system. The unfolded protein response system is a three-pronged system within the endoplasmic reticulum. And it's made up of these three transmembrane proteins, IRE1 alpha, PERK, and ATF6 alpha. And these three proteins are held inactive by a protein known as BIP, or the binding immunoglobulin. And so during periods of homeostasis, they're held inactive by BIP. Whenever there's endoplasmic reticulum stress, these transmembrane proteins become dissociated from BIP. BIP has a higher affinity to bind to misfolded proteins that result during ER stress. And so these transmembrane proteins then become transcribed, translated, to try and their goal is to try and tamper or stop whatever is driving ER stress and stress. And so in our work, we had really focused more on ATF6 alpha. And essentially what we were able to show was that what we demonstrated was that the envelope protein of HERV1 actually induces ER stress, and this was mediated through ATF6 alpha. Essentially, we were able to show an interaction between the envelope protein of HERV1 and ATF6, leading to cleavage of the alpha subunit of ATF6. And what usually happens is they are binding regions on RORC and STAT3 for ATF6. So RORC and STAT3 are the Th17 transcription factors. And what we were able to show was that this ATF6, following the interaction with HERV1, binds to the transcription promoter regions of RORC, STAT3 in Tregs, and then drove these Tregs to differentiate into Tregs that acquire a Th17 effector-like phenotype and produce IL-17. And as a consequence, these Tregs did not function normally. They did not suppress effector cell proliferation. And we know this because when we did silence HERV, we were able to recover some of that suppressor function of those Tregs. Now, together, these results suggested to us that HERV1, by inducing unremitting UPR activation, may contribute to failures in immunological tolerance by lowering the threshold for pro-inflammatory signaling in Tregs. Now, in spite of this, we know that there are several gaps that still need to be addressed. For instance, what does HERV1 RNA encode for? And what is actually being transduced? Now, the work that we had done, which I shared a bit of, really was done predominantly on peripheral blood regulatory T cells, or Tregs. So it's unclear if we still would have similar HERV upregulation in the liver. And even within the Tregs in the liver, would we see the same thing? And so we tried to look at this. And essentially, we obtained formalin-fixed, paraffin-embedded liver tissue from livers of transplanted children with alloimmune hepatitis, as well as similarly transplanted children who had graft quiescence and normal graft function. We isolated RNA from these FFPE liver samples and subjected that RNA to bulk RNA sequencing. And indeed, we did find upregulation of HERV in livers with alloimmune hepatitis compared to livers with immune quiescence and normal graft function. And this was confirmed on QRT PCR as shown here. Now, there is a lot in the cancer literature on what the specific HERV we had seen that was upregulated, specifically HERV-S71, does in the cancer literature. So essentially, we know that HERV-S71 encodes for full envelope protein that binds to a transporter known as the monocarboxylic acid transporter 1. And what MCT1 does is it catalyzes the transfer of lactate across from tumors to the circulation. And we know that actually this in the cancer world, it's actually become a target for treatment of cancer. Additionally, if you look at induced Tregs of cancer patients who are resistant to anti-PD-1 therapy, their Tregs have a higher MCT1 expression. And also, lastly, when you look at tumor infiltrating Tregs of patients who have worse clinical outcome, those tumor infiltrating Tregs have a higher MCT1 expression. And so this is work, of course, from the cancer world. And so this has led us to start this collaboration with the National Cancer Institute at the National Institutes of Health here, the NIH in the US. And we've been working with, and it's still in its infancy, we're working with two scientists, Mark Sidburn and Naomi Taylor, who have a tremendous amount of experience in looking at, their labs look at like transporters, receptors, and Mark Sidburn has a tremendous amount of experience with retroviruses in general. And so what they did was they looked at RNA-seq data, which I had incidentally when I was working at Yale. So the initial data I was showing you was some data that we collected when I was at Yale. And what they did was they've identified sequences of HERVs that are differentially expressed in our livers with alloimmune hepatitis compared to similarly transplanted livers with normal graft function. And these sequences that they have identified that are expressed in these livers are sequences that are likely bind solute carrier transporters that may have a potential significance in terms of metabolic regulation. And so one of the things that's important for us to determine is whether the whole envelope open reading frame of HERV-71 is eventually transcribed and translated in our patient samples. And to do this, we obviously have to look at our sequence reads, whether they cover the open reading frame, as well as using PCR or 3' 5' race on total RNA. Now, while this sounds all great, I do not have access to data as I'm no longer, I've left Yale now for about 7-8 years. And so what we've been doing is trying to put together a cohort at Georgetown. And so we're obtaining, again, formalin fixed paraffin embedded liver tissue from our same group of patients. I included patients with acute rejection just as another control. So these would be our children with alloimmune hepatitis, non-transplanted children with autoimmune hepatitis, similarly transplanted with normal graft function. And then we're going to isolate RNA and subject this RNA to long-read RNA and genomic sequencing. Additionally, we will use RNA, we plan to use RNA-scope and immunohistochemistry to assess the RNA of HERV sequences that are transcribed and differentially expressed in our livers with autoimmune hepatitis versus those livers with normal graft function. In addition to identifying cells, the cells that express these RNAs. Now, one thing that I wanted to share from here that we've been doing so far. So we at Georgetown, our center, that when livers are procured for transplants, we have a huge biorepository here. And so we obtain post-perfusion biopsies that are stored. And so what we did and what we have done is we asked the question, if we did see high HERV-71 expression in livers that were being transplanted, would it predict future development or future risk of developing alloimmune hepatitis after transplant? And so what we did was between 2021 and 2023, we went into a biorepository, obtained livers of livers that were subsequently transplanted into our pediatric patients from children whose parents had consented for research participation. we isolated both RNA and protein, and we looked at for expression levels of this HERVs71 in those livers. There were actually 16 children, but we got good samples from 15. We have already our patients here with alloimmune hepatitis, autoimmune hepatitis, and so they just served as our positive control. Their livers just served as our positive control. And so from our group of 15 livers that we looked at, so these livers have now been transplanted in our patients. We saw 3 livers had increased expression of HERVs71. We also looked at protein levels of HERVs71. And again, as you can see, even though there doesn't seem to be much discrimination within that group of livers that have been transplanted, the 3 patients who had shown the increased expression are listed as shown here in red. And essentially what we're doing is we sort of like we are following them to see if they will eventually develop autoimmune hepatitis. But, I just thought I should share this. So we'll see how that goes, if this would really be able to predict the ability to, if this would be able to predict the future risk of development of autoimmune hepatitis. We also have some ongoing work here with scientists at the Cancer Center at Georgetown, the Lombardi Cancer Center at Georgetown. And essentially what we're trying to do here is to try and identify cells that are expressing HERVs71 in the liver using imaging mass cytometry. And so we've used tissue that's formalin fixed paraffin embedded from our 3 groups of patients. And we're looking at generally the different types of cells, HERV expression in lymphocytes, monocytes, plasma cells, macrophages, NK cells, T follicular helper cells, as well as Tregs. And because of our interest in Tregs, we're also looking at the different phenotypes of Tregs in these inflammatory liver conditions to determine if they have anything, if they play any role in either curbing or prediction of response to therapy or lack of response to therapy. And so we're looking at for the presence of Th1-like Tregs, Th2-like, Th17-like Tregs, Tregs with that express this checkpoint engagement marker, suppressive type Tregs, phosphostat expressing Tregs, Tregs expressing exhaustion markers, or those Tregs that express a proliferating or an activation marker. And so what we have done is we've developed a panel of 32 markers, which we've used in our imaging mass cytometry. And these 32 markers are made up of transcription factors as well as proteins. And basically, we've put a panel that we've used to identify these different cell types. So these would be our lymphocyte panel. This is our monocyte macrophage panel, T follicular helper cell panel, naive Tregs with either suppressive phenotype or checkpoint engagement phenotype, Th17, Th1-like, Th2-like Tregs with an exhaustion marker, proliferative activation marker, and phosphostat Tregs. And so I have some very preliminary results from this imaging mass cytometry to share here today. Investigator actually, she was supposed to provide me with a bit more slides for the talk, but she unfortunately had to leave the country for a family emergency. So suffice to say, when we look at our comparisons between patients' livers with alloimmune hepatitis versus livers with autoimmune hepatitis, there appears to be an abundance of CD163 positive macrophages in livers with autoimmune hepatitis compared to livers with alloimmune hepatitis. And this is just another panel that tries to depict this. Now, these images are usually transferred bioinformatically into a graphical format. And so this graphical format is just showing what the images shows. Essentially, as you can see here, there is a trend towards an abundance of CD163 macrophages in livers with autoimmune hepatitis compared to livers with alloimmune hepatitis. We also see a trend towards an increase of Th2-like Tregs in livers with autoimmune hepatitis versus livers with alloimmune hepatitis. There seems to be a trend towards an abundance of Tregs with an exhaustion phenotype in livers with autoimmune hepatitis compared to livers with autoimmune hepatitis. And we do see in this case a significant abundance of Tregs with a proliferative marker in livers with autoimmune hepatitis versus livers with alloimmune hepatitis. Now, when we look at our comparisons between livers with alloimmune hepatitis to livers with normal graft function, so these are livers of patients with normal, similarly transplanted children, normal graft function, immune quiescence, we see a significant abundance of CD14 monocytes in livers with alloimmune hepatitis compared to livers with immune quiescence normal graft function. We also see a trend towards increased CD68 macrophages in livers with alloimmune hepatitis compared to livers with normal function, normal immune quiescence, normal graft function. Similarly, we see what appears to be a trend towards an abundance of Tregs with that checkpoint engagement marker, TIM3, in livers with alloimmune hepatitis versus those livers with immune quiescence, normal function, and an upregulation, again, increased abundance of Tregs with this activation type marker or antigen presentation type marker in Tregs with alloimmune hepatitis in livers with alloimmune hepatitis compared to livers with normal graft function. Now, in looking at this, one of the questions we asked ourselves was, can we, using image mass cytometry, can we be able to predict based on these different markers the response to medical therapy in patients who have alloimmune hepatitis? And we've been able to ask this question because we have 2 of our patients who had alloimmune hepatitis and then lost their graft, so we have access to their explant. And so these are really small numbers, by the way. But when we do look at the livers of children who have alloimmune hepatitis but are responding to medical therapy, and so at that point in time they had normal liver numbers, we see that there's a significant abundance of CD20 lymphocytes in their livers compared to livers of those children who clearly failed medical therapy and needed to come to transplantation. Similarly, in those livers of children with in those livers of children who are responding to medical therapy, we're seeing this we're seeing this significant abundance of Tregs that have this activated phenotype in their livers compared to livers of children with alloimmune hepatitis who failed medical therapy and obviously come to retransplantation. Now, on the next slide, I'm showing you they're not significant, but I share this just because there seemed to be quite a clear separation between the 2 groups. And so we're seeing this Th2-like Tregs, what appears to maybe there will be an abundance in livers that in alloimmune hepatitis livers that respond to medical therapy compared to those that do not respond to medical therapy. And what appears to also maybe if this holds up, an abundance of Tregs that express TGF-beta in livers of children with autoimmune hepatitis who respond to medical therapy versus those who fail medical therapy. Again, these are really small numbers, and we are hopeful that if we're able to get grant funding to make this a multi-center trial, we can increase our numbers and see whether or not these differences do indeed hold. And lastly, we see these are Tregs with a suppressive type marker, which we're seeing what would be an abundance in livers that respond to medical therapy versus those that do not respond to medical therapy. We try to do the same thing with our livers with autoimmune hepatitis as well. And we have only one patient with who did not respond to therapy and then was transplanted, and we had access to their explant. And what we did was we took the index biopsies of these patients, and the question we're asking is, if we looked at the index liver biopsy, are we going to be able to determine or predict who will respond to medical therapy and who will not respond to medical therapy? And that's really what we're trying to do here. And so you can see, I mean, we didn't put any statistics because obviously we have only one in this group, but there appears to be quite a separation with Tregs with a proliferative phenotype, almost seeing an abundance in those in the index liver of children who have autoimmune hepatitis and who are responding to medical therapy, compared to the liver of the autoimmune hepatitis liver that did not respond to medical therapy. And we see also what appears to be an abundance of CD163 and CD68 macrophages in those livers that are responding to medical therapy versus those that did not respond to medical therapy. But again, these are really small numbers, and we're really hopeful to get some funding to be able to make this a multi-center study, improve our numbers, and see if these differences will truly hold. We see again what appears to be what may be an abundance of CD14 monocytes in livers that respond to medical therapy versus those that do not. And then what appears to be abundance of NK cells in livers that do not respond to medical therapy compared to those that respond to medical therapy. So we've still got ongoing analysis of our imaging mass cytometry data, and we're also trying to look at spatial relationships between these cells. I think they usually call neighborhood analysis, but these are really slow bioinformatic analysis, but they're still ongoing. So I would just like to summarize it in my last few slides, that HERVs, we've reported and shown this, that they alter the immune response and Treg function in children with who have post-liver transplant autoimmune hepatitis. And we've shown that they do this through the interaction of its envelope protein with ATF6, inducing these Th17-like Tregs with loss of Treg suppressive function. And again, I put very like 3 times, preliminary results from our imaging mass cytometry suggest that autoimmune hepatitis may be characterized by monocyte-driven innate immune activation. And in contrast, autoimmune hepatitis may demonstrate this proliferative and Th2-like Treg phenotype alongside features of alternative macrophage activation. However, we still have several critical gaps in our understanding of immune pathobiology in autoimmune hepatitis and alloimmune hepatitis. And so there's still a lot of work that needs to be done. And with that, I'd like to end here, and I'm happy to take questions. Thank you.